the name in them, it usually is a really long name, but it ends with something like this, where it says ammonium chloride. I'm trying to see if I spelled that right. I don't know. I think that's right. And these are not halogens. You might be confused to think that they are because they've got chloride in there. Chloride would refer to just the Cl negative, okay? Whereas chlorine has no charge on it, or hypochlorite or something like that are true examples of halogens. In this case here, it's describing a salt. And the ammonium chlorides or the quaternary ammonium compounds, probably the best way to describe them is their surfactants or their detergents. Okay? These are now the most common antimicrobial additive to soaps. So as of maybe the past year, these have become the most common. And there's still question out as to whether do they really even work? Do they have an effect on improving or reducing bacterial numbers in hand washing? And that's actually something we're going to spend some time talking about on Friday in class. So we did an example of each of those. What about phenol? What are examples of phenols?
I had it in my
Yeah, and there are other examples of this too. Anybody have one of those? Anybody have like air diffusers at home that do like the essential oils? Those are phenols. Okay, so those are actually phenols. A lot of those lovely smells are some of the organic chemicals that these plants or other, I guess mostly plants, produce, and some of them actually have antimicrobial activity. Okay, so keep that in mind. That may be something we come back to later on. Okay, so chemical, one way to organize these. Did anybody have a different way to organize this information? Kelly?
We did disinfectants and antiseptics.
Okay, and I don't think you were the only one. I heard at least one other group do this too. So disinfectants and antiseptics. And most people did really well on this with the homework. But what's the major difference between the two of these? And I'll pick at random. I know most of you answered this question.
Whether or not it'll damage.
Okay, which one doesn't, typically?
Antiseptic.
Good. Antiseptics are safe to use on skin, whereas disinfectants are not. Now, a couple of things here. All antiseptics, we could consider them to be disinfectants as well. Generally, both of these groups, they will kill the majority of microorganisms, but not necessarily all. What oftentimes is most often missed or not killed with disinfectants and antiseptics? I know some people even put this in their information. For example, bleach works really well against all microorganisms. Or alcohol works really well against almost all organisms, except for what? Go ahead, Megan. Yeah, absolutely. Spores are resistant to many of these chemical factors. And so, actually, for example, alcohol, spores are completely resistant to alcohol. Put them in 70% alcohol, they will survive years and years and years and years and years. It does nothing to them. Whereas to a vegetative cell, we've talked about how like gram-negatives, for example, that alcohol takes away or dissolves that outer membrane, and so it weakens those cells. Most of these don't work against spores. Some do, not all do, though. Okay. Disinfectants, what would we put under disinfectants or antiseptics? So Kelly, Lauren, and Kylie, how did you guys?
Products, not like necessarily the chemicals.
Let's go for it.
So for the antiseptics, we put dish soap, hand sanitizer, rubbing alcohol, Listerine, and hydrogen peroxide.
Okay. H2O2, Listerine. Those were the three that I heard.
And then disinfectants, we just added Clorox and Bleach.
And this would largely be the same thing, right? Clorox is just the brand name for most of those. Other ones we could put under either of these categories? Are any misplaced? You could make an argument that maybe one or more is misplaced. And this argument is based on some of the similar factors or features that we talked about on Friday. We said that to kill a microorganism, it not only requires a certain amount of time, but it also requires what else? So like if I'm going to take E. coli and I'm going to throw it into, I don't know, 60-degree water. It needs time, but it also needs a certain degree or level of that stress as well. E. coli can probably survive a minute at 60 degrees Celsius. They can't survive 5 minutes at that temperature. Okay? So you could argue that, yeah, H2O2 actually is a good antiseptic. A lot of us use it on cuts or as an alternative on cuts. But that solution is a 3% solution. 30% hydrogen peroxide is not sold in stores because it is incredibly hazardous to our own cells. Okay? Hydrogen peroxide is an oxidizer. And so it takes nucleic acids and it changes the AGs, Ts, and Cs. It starts to change around how they look so that they get misread, leads to mutations. You could argue that bleach, it could be used as an antiseptic, right? In the case of like drinking water, for example, we put chlorine in drinking water as a way to, or in pool water too, I suppose, as a way to disinfect, but it's at concentrations that are low enough that they shouldn't cause much harm on us. Okay? So it can be context dependent here. There are cases in which a chemical, we can place it under one or more categories. I'd actually argue that H2O2 is one of those. H2O2 can also fit under what we call degerming agents. What's a degerming agent? What's the name implied? Get rid of germs. Hand washing. And hand washing is probably the most common example. In hand washing, how long are you supposed to wash hands for? 20 seconds. Sing happy birthday to yourself twice, roughly, is the rule of thumb, right? Or the ABCs, or twinkle, twinkle, little star, because it's the same tune, right? But in each of these cases, it's not that the amount of time of contact of the soap with the bacteria is enough to kill it. What you're really just trying to do is to get them into that soap so that they are washed away from your hands. It doesn't necessarily kill them in that 20-second period of time, but it's enough to remove them. Hydrogen peroxide can work in the same way. What happens when you put hydrogen peroxide onto a cut? It does burn. But what else? And that's the side of it that it's a disinfectant. It shouldn't hurt you. What's that? It bubbles. And the bubbling helps to bring up dirt, bring up bacteria from the depths of that cut. And so you could argue that that functions like a degerming agent. It's not, in that sense, it's not killing the microorganism, but it's helping to remove it from that wound. Okay? We could also argue dish soap fits under this category as well. Again, it depends on time of contact. Other ways that folks organize this information. Okay. I think I've covered the major things here. What's the most common target of any of these? These chemicals work because what do they do to a bacterial cell? They obviously kill it, but how? What's the most common way? Annie? Disrupt the membrane is a common feature. Degrade proteins is another one. Cause nucleic acid damage. Oftentimes it's disrupting something about the homeostasis or disrupting those macromolecules that we've talked about that lead to loss of function. And microorganisms, in most of these cases here, do not gain resistance to these chemicals. That's in contrast to antibiotics that we'll talk about later on, where we do see some forms of resistance taking place. Okay. Questions on the chemicals themselves, but more so, I'm more concerned with, do you guys have a clear understanding of the distinction between degerming, antiseptic, disinfectant, and then the one we haven't talked, which is a sterilant, or something that sterilizes? Any questions on those distinctions? Okay. Well, so how can you define effectiveness here? Okay, so the ring of clearness around that disc, yeah, would probably tell you something about effectiveness. So, Kelly, which bacteria did I give you? Megatherium, Bacillus megatherium. Okay, we'll just call it B mega. So which had the largest ring for E. coli? H2O2. How about for Bacillus? Which had the smallest for them?
Probably Lysol for both.
Lysol for both?
Yeah.
Did anybody... Go ahead.
So for E. coli was Lysol, and then 10% bleach for the...
Okay. How about the other ones? How about Pseudomonas and Staph epi? Which one had the largest zone of inhibition for each of those?
The Pseudomonas, the largest, was kind of tied between the 30% bleach and the hydrogen peroxide.
Okay. Who else had a Pseudomonas one? Similar or... Okay. Okay. So 30% bleach or H2O2. How about the smallest for those ones, Allison? Lysol? And then for Staph epi. Oh. Best? Worst one, Aaron? Lysol? Okay. So there's a few ways that we could look at this information, right? And when we say best here, we mean the one that at least has the greatest inhibition of growth. And if I'm understanding correctly, most everybody took that to mean the one that has the largest circle around it. What's actually happening there? So why did the bacteria not grow, for example, with the 30% bleach? Why didn't it grow closer to the disc? And this seems like a very not important question, and maybe it isn't, but I'm trying to make a point here. So why didn't the E. coli grow closer to the disc in the presence of 30% bleach or in the presence of H2O2? What's happening there in that area where there's a blank space?
Is it killing the bacteria?
It could be. Can we tell from this experiment that it's killing them?
No, because technically it could grow back. Like, I feel like if you let it sit for like a while, it could technically grow back.
Okay, so I'll let you. I let these sit at in the 30-degree incubator. These actually incubated for 72 hours. Okay, so you're thinking that it could grow back. It may be death. What else could it be? Alexi?
They're not growing?
It could just be that they're not growing, right? How would we be able to test that? So how could we actually test to determine whether they've died or whether the concentration is enough to inhibit the growth but not kill them? How could we figure that out? Annie?
Can you treat cells that have already been growing with it and then see if it kills them? Like if you were to put a disc on top of an already
Okay, so you're saying take a disc that has a chemical embedded in it, put it onto a lawn of bacteria, which this is, and then see if it kills them. Is that what, am I following correctly? How do you measure death, though? So think back to what we talked about on Wednesday last week. We did this. We looked at some sample data where we were doing measurements of. I think the experiment was you start with 1,000 cells per milliliter in different temperature conditions, allow them to incubate for 22, so on and so forth. You measure the growth over those different temperatures. How did we know there was death occurring? What would be true if we saw bacterial death? Exactly. Less cells than what you started with, right? So we can, and our eye is only able to detect maybe, gosh, we can probably only start to detect when there's about 10 to the 8th bacterial cells in a milliliter. A colony is about 10 to the 9th bacterial cells. Much smaller than that, and we can't detect it by eye. So how do we tell? What could we do to test for, in that area or in that zone, whether the bacteria are dead or not? How about this? What if we were to, let's say that you could take a swab of bacteria from that zone. Now what do you want to do? To test and see if it actually is alive or if it's dead. Think about this for a couple of minutes. I'm going to give you some time in your groups to think about this. But the question is, if you have a plate, here's your zone of inhibition around that, and let's say that you took bacteria right from inside this zone of inhibition, or this zone where there's no growth whatsoever. How do you know after taking the bacteria from that if they're alive or if they're dead? What could you do? And think about experimental techniques, or think about things that maybe we've even done in lab to measure growth.
Wait, you're asking if they were dead or alive?
Uh-huh.
If you take them from the zone of inhibition?
So how do you test if they're dead or alive?
Wait, I thought that area didn't even allow for growth.
It doesn't, but you don't know that they're dead, because no growth doesn't equal death. I haven't grown in height in 15 years. Am I dead? Maybe a bit on the inside. But as far as I know, I still have a heartbeat going.
Just like the bacteria?
Maybe. Bacteria don't have hearts, though. But they do have souls, and they cry out when they're killed.
Are they alive?
I don't know. You tell me. How would we test that? How would... I mean, the question is, how do you test if they're alive or if they're dead within that area where there's no growth?
You take a swab and you put it somewhere else.
Where do you put it?
On a different culture.
Okay, go with that.
Can you inoculate it?
Into what?
Those OB Brock tube things.
Great. What's going to happen if they're alive, Nancini?
They're going to grow.
And if they're dead?
And they're not going to grow.
There's your experiment. That's exactly what we would do. It's as simple as just taking them and putting them into a non-selective media, or one where there's not some sort of growth inhibitor. So if we were able to if we wanted to, what we could do is just take an inoculating loop, swipe that area, put it into some new LB. If they're alive, they'll grow in 24 hours. If they're dead, no growth whatsoever. Okay? These zones here tell us something about the chemical. We at least have been able to rank which one is best and which one is worse based on that zone. One of the things that we have to think about here is actually what's happening to that chemical in that zone. So I told you that at the middle of that, one of those discs, I'm going to draw this from the side now. Imagine you're looking at this from the side. There is 10% bleach right here at this source. What happens to the concentration as it radiates outward from that disc? Does it remain 10%? What happens to it? What was that? Sorry, I heard somebody say something. Okay, it decreases. Exactly. It's like as if you put a drop of food coloring into water. It diffuses out from that spot where it hits the water, right? And that's exactly what's happening here. The other name for this experiment is what we call a disk diffusion assay, or a disk diffusion experiment. It's also known as a Kirby-Bauer, named after the individuals who created it. So we have at the disk the highest concentration of chemical, and from there it diffuses out. There's a point at which the bacterial cells begin to grow. And that region right there is the lowest concentration of chemical that can still inhibit the growth of the microbes. That concentration is what we call the MIC, or the minimal inhibitory concentration. So it's the minimal concentration of a drug or a chemical that is still sufficient enough to block microbial growth. Molly.
So at that range, going out from that, are we going to see, so is it just like a hard stop basically, like even though the concentration is going down? Because like from the naked eye it looks like just nothing is going to grow in that range, even though it's like a lower number, even though it's like a spectrum. Do you get what I'm asking?
Yes, so you're saying like there is this continuous gradual decrease in the amount of chemical. Do you get a continual gradual increase in the amount of bacteria? That's a very good question. You might start to see this radiation of like no bacteria are dead to where you're moving out from that. Now you've got this zone where they're living, and then all of a sudden you see growth kind of pop up, right? Typically, this is not uncommon, where there's like this hard stop where there's above that concentration, it completely blocks the growth. Below that concentration, it completely inhibits it. There is another value known as the MBC, which is the minimal bactericidal concentration, and that's the minimal concentration to kill all microorganisms. And so in between the MBC and the MIC, you've got less and less death, and then at some point the concentration is low enough to where you now can start to visualize growth of the microbes. Can we tell what that concentration is on these plates? So, for example, with the 10% bleach, where that zone is, do we know what the concentration is where that zone of clearing ends? Allison, you're shaking your head no. It's correct. All right. We don't know. We have no clue whatsoever. One of the things to start to think about is how can we test that then? How can we figure out what is that exact concentration that inhibits the growth of the microorganism? Okay. And that's something that we'll cover on Friday. But let me come back to one more point. Compare the 10% and the 30% bleach real quick. So compare zones 2 and 3 with each other. Which one has a larger zone of inhibition? 3. Higher concentration of bleach leads to more effectiveness overall, right? Greater inhibition of growth. Again, two very important things that come into play in determining how effective is a chemical or a physical measure of growth going to be, physical or chemical control going to be at inhibiting growth. 1, time of exposure. 2, what? What's the second one? What was that?
Stress.
Stress. Or how much, right? The percentage, in this case, percentage of bleach. 30% bleach works better than 10% bleach. That shouldn't be surprising, but again, it brings up this point that the more you have of the chemical or the higher concentration of it, it's going to have a greater effect on inhibiting growth. Okay? Now, I think that maybe the last thing to point out here, and we'll come back to these plates again on Wednesday. Our reading for Wednesday, or the mini lecture for Wednesday, really kind of hits on these different types of techniques. But one of the things to point out is not all chemicals are equally effective against all microorganisms. And not all chemicals work against all microorganisms either. We saw in our list that actually in most cases, in most cases, hydrogen peroxide worked best. 3% hydrogen peroxide worked really, really well. And of those that worked least, Lysol most frequently worked least well, except for in the case of Bacillus megaterium. There's not one way that's going to work to inhibit the growth of all microorganisms. And so sometimes what we have to think about is the environment. Where are we trying to use these different control methods? For example, in a hospital setting. What are the microorganisms that we might most commonly worry about in a hospital setting? And I know some people in here work in hospital or clinical care settings. So what microorganisms are we most concerned with? Staph is a big one. Staph aureus is a huge one that we're concerned with. What else? That's it? We're just worried about staph? Okay. Using these chemicals is context dependent. Right? So sometimes if we're trying to inhibit staph, bleach is going to work really, really well. Will bleach work against spores of C. difficile? No. We've got to come up with a different method. Okay? So sometimes the choice here is going to be dependent on factors like, what does the patient actually have? Every room has to be, in some ways, disinfected and or sterilized when a patient leaves. So does the illness that the person has or any opportunistic infection that they've had, does that affect our decision? Okay, and that's how we have to start thinking about this. There's not one size that fits all for all microorganisms. Different chemicals are going to have different effectiveness based on the characteristics of the microorganisms. Right? All right, we'll stop there for today. Bring me back up the cards and the plates. I'll just stick the plates in the fridge and we'll use them again on Wednesday for a little bit more analysis. Have a great afternoon, everyone.